Okay, it should be recording right now. Um, and also if anybody else wants to record, I'm sure you should be able to. So here, great examples of hydrophobic interactions are lipids. As you mentioned, they have a hydrophilic head group, hydrophobic alkyl group. So hydrophilic head group, hydrophobic alkyl group. So when they interact with water, and yeah, you guys will be receiving the recording. I'll go over that at the very end. Um, so when coming into contact with water, water molecules become highly ordered. So what that means is, as you see here, they form a cage around the hydrophobic alkyl group in order to maintain their hydrogen bonds. So why is this important? They can't interact with the hydrophobic alkyl group. Water obviously can't interact with something that's non-polar. So, oh, okay. Water can't interact with something that's non-polar. So as a result of that, it still wants to maintain the hydrogen bonds it's creating. So it forms cages around the alkyl group. And as you see here, um, this is how it works in increasingly ordered molecules. So you see here, each lipid molecule forces surrounding hydrogen molecules or water molecules to become highly ordered. However, what happens if you have a cluster of lipid molecules? As you see here, only the ones at the edge of the molecule can create a strong ordering of water. However, in the middle, there isn't as strong of an ordering, so entropy is increased. And there's also something known as micelles. So what are micelles? Micelles are when the hydrophilic head groups basically surround the hydrophobic alkyl groups such that it creates almost like it creates a sphere in and of itself so that the hydrophobic alkyl groups only interact with each other. Nothing can interact with the hydrophobic alkyl groups because the head groups form a sphere surrounding it. And so entropy is just energy and disorderness. So disorderness is further increased because water obviously cannot interact with what's in the middle. So now we're going to talk about cellular environments and tonicity. So I believe you guys did an osmosis lab as well. So in an isotonic solution, there is no net water movement. Oh, sorry. In an isotonic solution, there is no net water movement. It means that extracellularly and intracellularly, the amount of solutes on the outside of the cell is equivalent to the amount of solutes on the inside of the cell. In a hypertonic solution, there are more solutes outside the cell, such that water moves out of the cell and the cell starts to shrink. In a hypotonic solution, it's the opposite. There's more solutes inside the cell, so water moves into the cell. If water moves into the cell starts to uh, swell up, and eventually, due to the amount of water that's coming in, it can burst. So isotonic, no net movement. Hypertonic, water moves out the cell. Hypotonic, water moves in the cell. So acids and bases. Acids and bases are extremely important because of the fact that you'll learn about them in buffers as well, and hydrogen ions as well are extremely important in all chemical systems of our body. So there are things known as weak acids, weak bases, strong bases. So a weak acid is an acid that can partially dissociate into hydrogen ions and its acid component. So here, an example is acetic acid, CH3COOH can dissociate into CH3COO minus and then an H plus ion. So it's partially dissociated. However, a strong acid completely dissociates, such that there are no hydrogen ions with the acid. So A just resembles any strong acid, so it could be Cl, H2SO4, can fully dissociate into hydrogen ions and chloride ions, or hydrogen ions and SO4 ions. A weak base is, well, the opposite. Weak base is, well, it amines, for example, for amines, they can partially combine. So hydrogen ions and any base can partially combine. However, strong bases, they fully combine, such as sodium and OH, forming NaOH. So the reason for strong acids and strong bases, the reason why they're important is for buffers as well. So what specifically are buffers? Buffers are weak acids. So they have an ability to ionize partially and they're reversible. So here an example of a common buffer, bicarbonate. Carbonic acid breaks down into bicarbonate and H ions. So why do we want to use a weak acid rather than a strong acid for a buffer? So When Here's an example for NaOH. So NaOH is a strong base. When it reacts with carbonic acid here, it breaks down into bicarbonate and then water. So bicarbonate retains hydrogen ions. Because bicarbonate retains hydrogen ions, it makes sure there isn't a lot of free floating hydrogen ions, such that if there's a lot more free floating hydrogen ions, that can change the pH significantly. However, because bicarbonate retains some of the hydrogen, it's making sure that the free floating ion concentration is relatively unchanged. As a result of that, it can maintain an equilibrium that can neutralize any strong bases or strong acids because it doesn't dissociate completely. If it dissociates completely, for example, here, it would increasingly go to more basic. However, because there's a buffer, it maintains some of the hydrogen ions so that any large quantities of base, the buffer makes sure that there's a relatively small change in pH. So here is what's known as the midpoint, where the buffer pH is equivalent to the pKa. So it's the point where half of your weak acid is titrated with half of the weak base. Typically, any buffer range is about +1 and -1 pH units above or below the pKa. So as you see here, the pH and the pKa is 5. So +1, 4. Oh, sorry, +1, 6, -1, 4. So that's the buffer range is typically +1, -1. And then pH is equal to negative log of your hydrogen ions. So now what are isomers? Isomers are molecules that have the same chemical formula, but their atoms are arranged differently. So there are several types of isomers. There are structural isomers, as you see here. Structural isomers have constituent atoms that join differently, and as a result of that, have different chemical properties. So as you see here, butane is just 4 carbons in a row. Isobutane is 3 carbons in a row with another carbon bounded to the second carbon. So it still has the same structural formula. If you see here, 1, 2, 3, 4, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So it's C4H10. It'll be the same here. 1, 2, 3, 4, C4, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So it still has the same exact chemical formula, but because the atoms are arranged differently, it has completely different chemical properties. Then there are optical isomers, as you see here. Optical isomers are mirror images of each other. So optical isomers can only occur if there are 4 different atoms or groups attached to one carbon. So this is the carbon molecule here. In order to have an optical isomer, each molecule that's bounded to it has to be completely different. If any two of these molecules are the same, it cannot form an optical isomer. So they could definitely try to trick you on with that on the test. It could ask you what's the property of an optical isomer. The main property, 4 different groups have to be bound to the carbon. Then there are also cis and trans isomers. Cis isomers, so for cis and trans isomers, that has to be revolving around a double bond. So as you see here, there's a double bond between 2 carbons. When it comes to cis isomers, the double bond, any carbons or any molecules that are attached to the double bond, they're facing the same side. So as you see here, for cis butene, both carbons are facing the same side. For trans butene, it's opposite sides. So cis, same side, trans butenes, any molecule that's attached to the double bond, they have to be facing on opposite sides in order to be a trans isomer. Oh, yeah. Okay, so first knowledge check. As I mentioned, you can respond in chat, respond directly to me. Uh, yeah, the atomic number of an element. What is that? Okay, we've got a lot of answers. So the atomic number of an element is, yep, B. So atomic number of element is the amount of protons in an atom. So hydrogen has one. So the atomic number of hydrogen is one, and so on and so forth. Which of the following statements about the isotopes of an element are not true? Okay. Nice. So everyone put C. That's correct. So they all have the same number of neutrons. That's not true. I know when I asked this before, some students put E. They all have identical chemical properties because they thought, oh, they don't have identical chemical properties. But that's not about isotopes. That's about isomers. So if the question asked which of the following statements about isomers is not true, then it would be E. Um, Here, a last knowledge check for chapter 2. Okay. Everyone got it correct. Yeah, so it's the formation of ions by dissociation of an acid. What helps is that obviously knowing that HCl is a strong acid, they could ask for NaOH, which would be a strong base, etc. So now we're going to chapters 3/4, which is about macromolecules as a whole. So it's a pretty dense chapter. So a recurring motif in biology is that the structure of a molecule is equivalent to its function. As we learned about isomers, different structures of an isomer can create a different function. So the basic premise of macromolecules, you have different subunits, which are sugars, amino acids, nucleotides. They form covalent bonds within one another, creating macromolecules, which are your proteins, RNA, DNA, carbohydrates, etc. And then eventually, these macromolecules can combine with other macromolecules to form macromolecular assemblies, such as a ribosome molecule. So in order to make up a polysaccharide or sorry, not a polysaccharide, a polymer, you need a monomer. A monomer is just a single building block. So monomers are building blocks of macromolecular polymers such as sugars, amino acids, and nucleotides. And then these monomers combine with other monomers to make up a polymer, which is the macromolecular form. So sugars can combine with other sugars to create a polysaccharide. Amino acids combine with other amino acids to make proteins, nucleotides to make nucleic acids. So, how are things joined in together? So, the way things are joined in together are through condensation reactions, in which a water molecule is removed. So, condensation reactions are also known as dehydration reactions. So, each monomer is connected to a hydrogen ion on one side, OH molecule on another side. What essentially happens is that this OH molecule attaches to this hydrogen molecule of another monomer, removing water from both monomers. So now that OH and H is gone, the monomers are free to create a covalent bond within one another. And then this keeps on going in order to keep on forming uh, more monomers, in order to keep on combining more monomers and then create polymers. So in order to create polymers, condensation reactions, which means releasing water, removement of water. So the opposite is also true when you want to break down polymers into monomers. So in order to break down a polymer, you add water such that the water molecule breaks down into its hydrogen ion and OH molecule so that the OH molecule can bind to one monomer, hydrogen ion can bind to the other molecule, monomer, and then it can keep breaking down by adding more and more water. It can keep breaking down the covalent bond. So hydrolysis reactions, hydro, water, lysis is breaking down. So hydrolysis reactions break down polymers into monomers. Condensation reactions create polymers from monomers. So lipids. Lipids are extremely important. This is an example of a triglyceride. Triglyceride is synthesized from a glycerol, which is an alcohol, and three fatty acid molecules. This is just showing what I mentioned. Through a condensation reactions, OH molecule of this carbon here can take an H molecule from this oxygen here, releasing water, thus creating an ester linkage between the glycerol and the fatty acid molecule. So phospholipids are extremely, extremely, extremely important. Why are they important? They're the building blocks of every biological membrane in our body. So what is a phospholipid? A phospholipid contains a phosphate derivative here, sorry, phosphate derivative here, linked to two fatty acid chains through a glycerol. Now, it is also known as amphipathic. So, amphipathic means that it has a polar region and a nonpolar region within the molecule itself. So, the polar region of the molecule is this hydrophilic head. This hydrophilic head can freely interact with water and any liquid environment. The nonpolar hydrophobic tail cannot interact with water. So thus it is amphipathic. It contains two regions that are polar up top, nonpolar at the bottom. And so why is this important? This creates what's known as a phospholipid bilayer. So phospholipid bilayers have a hydrophilic head that interacts with the extracellular environment as well as the intracellular environment, while the hydrophobic tails can control what ions, what molecules are able to pass through the phospholipid bilayer. And before we talked about micelles, uh, the phospholipid bilayer creates a micelle in and of itself, such that all the fatty acid tails are only in the middle, while the hydrophilic heads are the only ones on the outside. So now we will talk about sugars. So when we talk about sugars, monosaccharides are often referred to as sugars, and glucose is the most common form of sugar. So, uh, when you see here, you have cellulose molecules, starch, and glycogen molecules. Monosaccharides, such as glucose, can combine to form cellulose, and then they can also combine to form starch and glycogen. However, different linkages, as you mentioned before, different linkages can have different effects. So you see here, or what we mentioned before, the structure can change the function. As you see here for cellulose, cellulose is very linear, it's in a straight line. However, starch and glycogen have different linkages such that it creates branches, and these branches change the structure, which ultimately changes the function. So as you see here, polysaccharides can also be modified in order to create special functions. So you have a fructose molecule here, and fructose molecules can have two phosphates added to it. So this molecule then becomes fructose 1,6 bisphosphate. Eventually, you're going to learn about cellular respiration, which is how our body takes energy from these glucose molecules, from these carbohydrates, and creates ATP to get energy for us. So fructose 1,6 bisphosphate is extremely important because it's one of the main molecules responsible for obtaining energy from glucose and then converting it to ATP. So by modifying fructose to add two phosphates, now it's involved in cellular respiration. As you see here, you have amino sugars. So amino groups here, the monosaccharides, glucosamine and galactosamine are amino sugars. They have an amino group in place of a hydroxyl group. A hydroxyl group is an OH group, and by adding that, it changes the function such that, for example, galactosamine is responsible for being a part of cartilage, which is a connective tissue in vertebrates. So by adding an amino group now, the sugar is now used for connective tissues. Then here's another example about chitin. So chitin is a polymer of N-acetylglucosamine. So what these N-acetyl groups do here is that these N-acetyl groups provide additional sites for hydrogen bonding between polymers. So why is that important? Why is it important to create more hydrogen bonding? Well, chitin is the external skeletons of majority of insects. And chitin needs to create a strong protection for these insects when making these exoskeletons. So by creating more hydrogen bonds, which is a strong bond in and of itself, by creating additional sites for hydrogen bonding, it makes it that much stronger so that the external skeletons of the insects can be safe. Now we're going to talk about polypeptides. So polypeptides have an amino acid, which is an aminated carboxylic acid. You have a carboxyl group here, a side chain, and the alpha carbon here. So I think you guys did this in a homework about different side chains, like what makes up an amino acid. Every amino acid has the same exact structural backbone. What that means is that every single amino acid you'll see has the same exact structural backbone of an amino group, alpha carbon, and a carboxyl group. Every single one of them. The only difference that changes each amino acid's structure and function is the R group, which is the side chain. So the side chain changes each, oh, sorry, the side group changes each and every amino acid structure and function. However, other than the side chain, they all have the same exact backbone. So for polypeptides, as you mentioned before, the polypeptide backbone consists of repeated peptide bonds between the amino group and carboxyl group. Monomers are added at the carboxyl terminus here. So monomers are added at the carboxyl terminus, where once again, condensation reactions, water leaves, and the polypeptide grows from the amino group. So the polypeptide grows from the NH3 group to the carboxyl group. So anytime any new amino acids are added in order to create a polypeptide, it will be added to the carboxyl group. So when it comes to cysteine disulfide bridges, um, cysteine disulfide bridges are extremely important because they help create a stronger bond between sulfur molecules. So when you have cysteine molecules in polypeptide chains, here you have so cysteine amino acids have side chains that contain sulfur. And what these do is that they can release hydrogen so that the sulfur molecules can create a bond within itself. And these disulfide covalent bonds help create just a much stronger and stable molecule. So, as we mentioned before, polypeptides make up amino acids, and these amino acids can make up many different structures. So you have primary, secondary, tertiary, and quaternary. So primary structure that defines the protein identity. So each amino acid that's just in that order, the order of the amino acids defines the protein identity, whether it's a hemoglobin molecule, myosin molecule, albumin molecule, etc. They all differ in their amino acid sequences. You kind of think of a primary structure as just the name, like creating a name for the amino acid. So if you change one of the amino acids in the structures for another amino acid, it completely changes the name, so it completely changes the protein itself. It could even get rid of its function as well. So we'll talk about mutations later on, but the order of the amino acids is extremely important as that helps create the identity of the protein. However, several proteins of the same type have similar amino acid sequences across species. For example, the insulin protein sequence is very similar across different mammals. So even though humans, monkeys, whales, etc., even though all of us use insulin in different ways, the insulin protein itself has very similar sequences. And as I mentioned before, modifications in the sequence can have extremely dramatic functional consequences. Then, after the primary structure, you can take two polypeptide chains and then they can create secondary structures. And these secondary structures can occur in two different ways, an alpha helix or a beta pleated sheet. For in an alpha helix, the hydrogen bonds form between the amino group and the carboxyl group. So hydrogen bonds form between the N group, NH hydrogen, and then the carboxyl group, the CO oxygen group. So these hydrogen bonds can help stabilize the polypeptide backbone into a helical structure here. And then any side chains, which are the R groups, any side chains project away from the core of the helix. Then beta pleated sheets are when hydrogen bonds form between the carboxyl and amino groups between the polypeptide backbones. And what this does is that the arrangement stabilizes multiple chains of polypeptides into creating this sheet. Next, you have a tertiary structure. So a tertiary structure is where R group interactions, so the interactions between various R groups can fold it into its tertiary structures. And as you mentioned before about the disulfide bridges, why that's important, disulfide bridges can help stabilize these tertiary structures. And as you see here, you can take a beta pleated sheet, which is one of the secondary structures, and an alpha helix, which is another secondary structure, combine those to create a tertiary structure. And in order to stabilize it, you have the disulfide bridges. And then lastly, this can create one subunit. A quaternary structure is many tertiary structures essentially combined. And so, in order to stabilize the association among all of these different polypeptides, you have hydrogen ionic bonds, hydrophobic interactions, as well as disulfide bridges once again to help stabilize all four of these subunits coming together. So, as I mentioned before, hydrophilic and hydrophobic interactions help stabilize the 3D structures of polypeptides. So polypeptides that have hydrophilic side chains tend to remain in the cytoplasm. They usually assume a linear configuration here. However, polypeptides that have a hydrophobic side chain, they want to be in hydrophobic environments. So where is a prime hydrophobic environment in the cell? The cell membrane, typically inside the cell membrane. So polypeptides that have these hydrophobic side chains want to be embedded within the cell membrane itself. So this is just showing what we talked about before about interactions with the environment as well as like micelles. They tend to fold into spherical structures, which is stabilized by hydrophobic interactions. Obviously, as you see here, if the outside of the environment is hydrophilic, then the hydrophilic side chains want to be on the outside, making sure that the hydrophobic side chains remain on the inside. As mentioned before, this can create a micelle here. So you have an unfolded polypeptide, you have nonpolar side chains and polar side chains, and then they create a sphere. And when they interact with the aqueous environment, they create a sphere such that there's a hydrophobic core region that contains nonpolar side chains, as well as hydrophilic side chains forming on the outside. And that allows for hydrogen bonds to form on the outside as well. So as you see here, many different protein structures. Um, and so these protein structures there's several different types of proteins. So they're not going to test you on what is the function of this specific protein. So you're not necessarily going to have to memorize all of these proteins. However, the function of the protein is in the name. Let's see here, receptor proteins, they're receptors, they respond to chemical signals such as ligands later on. Signaling proteins help control processes. You have defensive proteins such as antibodies that can respond to pathogens, structural proteins, storage proteins, etc. It's within the name itself. So as long as you could recognize like what the name means, then you can have a general idea of what the function is. So now we're going to talk about polynucleotides. So how are polynucleotides formed? Polynucleotides are formed from a base. A base can either be a pyrimidine or a purine. Your purine contains two rings, adenine and guanine. A common mnemonic that people use is pure as gold. So your purines are adenine and guanine, and you have your pyrimidines, which are cytosine, thymine, and uracil. Uracil particularly found in RNA. So these are your bases. Then you have a ribose or a deoxyribose, as well as a phosphate. So what's the difference between ribose and deoxyribose? One thing you'll learn in biology, prefixes are extremely important. Deoxyribose obviously means it does not contain an oxygen. So in ribose and deoxyribose, if you look at the second carbon here and the second carbon here, in ribose, the second carbon is bound to an OH molecule. However, in deoxyribose, the second carbon is only bound to an H molecule. There's no oxygen present here. So that's why it's referred to as deoxyribose. And so adenine can bind with thymine slash uracil. Guanine can bind with cytosine. So how are polynucleotides made? Polynucleotides are made once again through condensation reactions. However, this reaction occurs in a specific manner. So the specific manner is that it forms from the 5' carbon to the 3' carbon. Any nucleotides are added to the 3' end of a growing polypeptide chain. So what does 5', 3' mean? As you mentioned before, uh, as you see here, it is a 5 carbon sugar. The 5 carbons are 1, 2, 3, 4, 5. So any new nucleotide is added to the 3' end here. So a new nucleotide is added to the 3' end where the phosphate group of a nucleotide attaches to the 3' end of the carbon. Same thing here. The phosphate group of added nucleotide will combine to the 3' carbon of the nucleotide, releasing water through the condensation reaction. So elongation which is the term for creating more, which is the term for creating a polynucleotide, elongation proceeds from the 5 prime direction to the 3 prime direction. So anytime a new nucleotide will be added, the phosphate group will bind to the 3 prime carbon. So what's the differences between RNA versus DNA? So the main thing you we want to go over is that first, the bonds. So adenine and thymine create hydrogen bonds within each other, and then cytosine and guanine create hydrogen bonds within each other. So adenine and thymine create two hydrogen bonds, cytosine and guanine create three hydrogen bonds. So this is in DNA. However, in RNA, adenine binds with uracil instead of thymine. Cytosine and guanine remains the same in both RNA and DNA. However, in RNA, adenine binds with uracil. Furthermore, RNA is typically single-stranded. It's very rare to find double-stranded RNA, whereas DNA is always double-stranded. So adenine binds with uracil and RNA, and RNA is typically single-stranded, while adenine binds with thymine and DNA is typically double-stranded. Furthermore, adenine and thymine have two hydrogen bonds, cytosine and guanine have three hydrogen bonds. Furthermore, there's something known as Chargaff's rule. Chargaff's rule is that the amount of adenine is equivalent to the amount of thymine, which makes sense if it's creating a bond with thymine, and the amount of cytosine is equivalent to the amount of guanine. So a question could ask, if there is 30% cytosine in a DNA molecule, what percentage adenine is there? So if you do the math, if there is 30% cytosine, that means there's also 30% guanine. 30 plus 30 is 60. You have 40% left. That 40% must be split evenly between thymine and adenine. So 40 divided by 2 is 20. So you have 20% adenine. So that's Chargaff's rule. C is equivalent to G, T is equivalent to A. If they ask for Chargaff's rule for RNA, it is the same thing. A is equivalent to U, C is equivalent to G. So this is just a general table distinguishing RNA from DNA. As I mentioned before, RNA has ribose, which does contain an oxygen. It's ACGU or AUCG if we're going by pairs, and it's usually single-stranded. DNA does not have an oxygen on the second carbon. It's ATCG, which are the pairs, and it's double-stranded. So, which of the following statements about the primary structure of a protein is not true? Okay, two different answers so far. This is, oh, three different answers so far. hmm All right, so this is definitely a hard question. So the correct answer is actually not C. The correct answer is A. It may be branched. If we go back, here. So if we go back, as I mentioned before, when it comes to primary structures, so the order of the amino acids determine the primary structure. Therefore, it is unique to whatever protein is trying to make. For example, albumin, if any one of these amino acids here is different, then it's not going to become albumin anymore. Or let's say insulin. If this, let's say this structure or this order of amino acids makes insulin. If any single one of these guys here change, then you're not going to be able to form insulin anymore. Therefore, it is specific to that protein. The order of the amino acids is extremely important in determining what protein it will become. And any modifications can have extremely dramatic functional consequences, such as getting rid of the function as a whole or even completely changing the protein itself. And another thing, primary structure is just the order of the amino acid. Therefore, a primary structure cannot be branched. It is specifically just a single line. You can kind of imagine it as a single line where you're creating letters. That's why I mentioned the name. Like each amino acid you can think of as a letter. You're just creating the name of whatever protein you're trying to make. So the correct answer was I believe A. Yeah. The correct answer was A. So the primary structure cannot be branched. And as mentioned before, it does determine the tertiary structure and it is unique to whatever protein. Question two, the amphipathic of nature, what does that mean? Or what's its importance? All right. Yeah. So the correct answer, it is B. It is important in membrane structure. As I mentioned before, why is it important in membrane structure? You have a polar region, which is the hydrophilic head, interacts with the extracellular and intracellular environment, and you have the hydrophobic region, the nonpolar region, which makes up the tail and controls what ions and molecules are going through. What is a nucleotide made of? This should be fairly simple. Oh, a nucleotide in DNA. What is it made of? Okay, we have differing answers here. Some people are saying E, some people are saying B, some people are saying C. The correct answer is C. So a base plus a deoxyribose sugar plus a phosphate. So to the people that said E, almost there, but here, let's just go back. I know I didn't mention it that much, but there is a phosphate nucleotide. And wait, no, I did mention it because the phosphate group of the nucleotide is what attaches to the 3' carbon when you elongate a molecule. So, a nucleotide in DNA contains a base, a deoxyribose in DNA, and then it also contains a phosphate. And this phosphate is extremely important because this phosphate is what attaches to the 3' end and when you elongate a nucleotide molecule. Um, oh, why couldn't it be here? Uh, sorry. This question. Okay, so someone asked for this question, why can't it be C? It's because what this question is saying that phospholipids are polar and they're not non-polar, but they are non-polar. They have a polar side and a non-polar side. So this is like certain answer choices try to like trip you up just with the wording. So here I could kind of see where the confusion is with answer choice C, where you think it's saying that it has both regions, but here the answer choice is saying that it only contains a polar region and it does not contain a non-polar region. But that's not true. The amphipathic nature means it contains both a polar and a non-polar region. So you gotta, one thing you have to make sure of is that when you read the question, read each answer choice and the wording carefully because sometimes they try to trip you up like here, where you think it means it contains both, but in reality, the answer choice itself is saying it only contains one of it. As I mentioned before, nucleotide is C. Uh, don't forget about the phosphate. What is the difference between DNA and RNA? All right, so yep, the correct answer is C. Um, Once again, someone mentioned C and A, but they changed it later on to C, obviously. But this is what I mean that wording is extremely important. Take your time to read each answer choice carefully. And another thing is the test is almost like an endurance exam as well. You're going to be going through around 8 to 10 questions per chapter. So anywhere from 50 to even 70 questions. Sometimes it could be 8 to 14 questions per chapter. So, like I mentioned, anywhere from 50 to 70 questions. So, it's an endurance exam as well, in that you have to make sure you read each answer choice carefully. A, you could read through it fast, think that it says DNA is double-stranded, RNA is single-stranded, and choose that answer instead. So, the correct answer is C. DNA contains deoxyribose, RNA contains ribose. Why isn't it B? So no, whenever you see something say only, it's probably not the answer because only limits what it's able to do. So DNA is not only informational. DNA can also be catalytic. Same thing for RNA. RNA can also be informational because there are let's say viruses such as retroviruses which use RNA to create which has that information in order to create more virions, which create more viruses, etc. So whenever you see an answer choice with only, typically that won't be the answer because it's limiting what its function is. So DNA can be both, RNA can be both. All right, now we're moving on to chapter 5, the cell. So chapter 5, 6, 7, and then we'll be done. So surface area to volume. Why is this extremely important? You always hear that, oh, you want an increased surface area to volume ratio in your cells. So the reason for that is that volume determines the amount of chemical activity in the cell per unit time, while surface area determines the amount of substances that can pass through the cell boundary per unit time, such as getting rid of waste or influx of oxygen and nutrients. So having a greater surface area to volume ratio, the cell will be more and more efficient at material exchange. So now we will be talking about prokaryotes and eukaryotes. So what are prokaryotes? Prokaryotes include all bacteria and all archaea. So prokaryotes are extremely unique. Do we have to memorize that? Memorize which thing? The the surface area to volume ratio? No, you don't have to memorize like the surface area formula or the volume formula. I don't think you have to memorize that. I think the most important thing to memorize is that a greater surface area to volume ratio means that there will be more material exchange that's occurring. So this guy will have a lot more material exchange that's occurring because it has a much greater surface area to volume ratio. All right, so as I mentioned before, prokaryotes include all bacteria and all archaea. So they're extremely unique as they are unicellular organisms and they don't contain any membrane-bound organelles. So they have what's known as a capsule. A capsule is a protein coat that protects the prokaryote. They do have a cytoplasm, they do have ribosomes and a plasma membrane, and they also have a nucleoid. So their nucleoid is similar to our nucleus region, except their nucleoid region is just a single DNA molecule. That's it. It's not as compact and not as what's the word? Not as complex as extracell as eukaryotes. They also have a flagellum. Flagellum is extremely important because flagellum helps move the prokaryote through uh through its environment. Now we have eukaryotes. So eukaryotes do contain membrane-bound organelles. And yes, you will be responsible for knowing every single function of everything that's written here. Trust me, it is not that bad. So first things first, you have the nucleus region here. The nucleus region contains all the DNA of your cell. The nucleolus region is responsible for creating ribosomes. So nucleus contains your DNA, nucleolus contains ribosomes. This all seems so hard to remember. It is, I mean, it is a lot of material, but the functions aren't that difficult. It just looks like a lot, but trust me, it's not that difficult in terms of what its function is. The function itself is very simple. So nucleus contains the DNA. Nucleolus makes ribosomes. Cytoskeleton is extremely important as that maintains the structure of your cell. You have peroxisomes here. Peroxisomes break down lipids through hydrogen peroxide. You have your Golgi apparatus. Your Golgi apparatus is responsible for packaging and sending out vesicles, which contain proteins, etc. Um, so like I mentioned, peroxisomes break down lipids through hydrogen peroxide. The Golgi apparatus itself um, packages proteins and lipids and then sends them out into the cell or out of the cell through vesicles. Obviously, your plasma membrane acts as your cell membrane. Mitochondria, like always, people always say, powerhouse of the cell. As long as you know that mitochondria is where cellular respiration occurs and where ATP is made. Now you have your smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth ER makes lipids. Rough ER makes proteins. As simple as that. So if the question asks what the smooth ER does, it is responsible for making lipids. The rough ER is responsible for making proteins. And then the Golgi apparatus is the area where after lipids and proteins are made, it goes into the Golgi apparatus, which packages it and then sends it out, either to another area within the cell or extracellularly as well. So eukaryotes and plant cells are extremely similar. However, in plant cells, it also contains a vacuole, which is a water-containing region. It also contains chloroplast, which is responsible for photosynthesis, and plasmodesmata, which is responsible for sending out nutrients within cells. All right, so membrane cycling. So what membrane cycling is, membrane cycling just means that materials are transported within the cell through several methods. So first, lysosomes. Lysosomes are created from the Golgi apparatus. So then a lysosome is sent out. Lysosomes can then fuse with phagosomes. And what are lysosomes? So digestive enzymes, which help digest any material within the cell, outside the cell, etc. Digestive enzymes are concentrated within the lysosome and then they're pinched off. Then there's phagocytosis. Phagocytosis is when food particles are taken in by the cell. So this is the outside of the cell. If there are any food particles outside of the cell, it can be brought within the cell through a method known as phagocytosis. So now you have your lysosome, which is responsible for digesting. Now, and then you have your phagosome, which is just a I wouldn't call it a vesicle, but the phagosome is just a membrane that brought in food from outside the environment. The lysosome can then fuse with the phagosome such that now your digestive enzymes can obviously digest the food that was brought in extracellularly. So small molecules that were generated by digestion diffuse within the cytoplasm, and any undigested materials are then excreted through the cell through exocytosis, which is essentially what happens. This secondary lysosome here binds to the cell membrane and then it just releases its content outwards. So as I mentioned before, the cytoskeleton helps maintain the structure of the cell. So what that means is that it helps maintain and support cell shape, provides support for cell motility. Motility means movement, and it also provides scaffolding for intracellular transport. So you have your microfilaments, which are made up of actin and often interact with strands of other proteins. So for microfilaments, just think of your muscles. Any muscle movement is due to microfilaments. You have your intermediate filaments here, which are made up of fibrous proteins that have rope-like assemblages, and they help stabilize the structure and maintain shape. So intermediate filaments are also extremely important. Some common examples of intermediate filaments are keratin and desmin. Next, you have microtubules. So microtubules are responsible for providing the scaffolding for intracellular transport. So microtubules are extremely long, hollow cylinders that are made up of tubulin, and there are motor proteins known as kinesin and dynein, which help move it along microtubule tracks. So here's an example of kinesin and dynein. So you have your microtubule here. As I mentioned before, for microtubules, extremely important for your intracellular transport. You have your kinesin here, which is contains a vesicle that's transporting it, and then essentially it just walks along the microtubule. So it detaches part of its the kinesin detaches part of it and then reattaches the other side, and it keeps on doing that, and in a way it walks along the microtubule, helping transport molecules. So, oh, just to go back here. So kinesin moves particles from the minus end to the plus end of the microtubule. So next you have your mitochondria. Your mitochondria helps convert fuel molecules into ATP. What's typically your fuel molecules? Your glucose. Mitochondria contains two lipid bilayers. It contains an outer membrane and an inner membrane. The space between the two membranes is known as the intermembrane space, and then the space enclosed by the inner membrane is known as the matrix. So in order from outside to inside, it goes outer membrane, inner membrane space, intermembrane space, inner membrane, and then the matrix. And then what your cristae are just the folds caused by the inner membrane protruding into the matrix. So these folds are known as cristae. So here you have chloroplasts and plant cells. So as I mentioned before, mitochondria is responsible for cellular respiration, which makes ATP in animal cells. In plant cells, you have chloroplasts. Chloroplasts convert light energy into chemical energy. So chloroplasts contain two lipid bilayers. They contain an outer membrane and an inner membrane. So the inner membrane here, the inner membrane surrounds the stroma. So it surrounds the stroma here, which is responsible for structural support as well as nutrient and gas exchange. The stroma also contains what's known as the thylakoids here. The thylakoids are responsible for the light-dependent reactions of photosynthesis. You don't need to know what the light-dependent reaction itself actually is yet. You'll need to know that for the next exam. Just know that the thylakoid is responsible for one of the reactions of photosynthesis and the chloroplast as a whole. So what is the endosymbiotic theory? The endosymbiotic theory posits that some intracellular organelles, such as mitochondria and chloroplasts themselves, were once independent, free-living prokaryotic cells, and then eventually they were incorporated into larger cells. So the reason for this belief is that both mitochondria and chloroplasts are self-replicating organelles. So they're able to replicate by themselves. They also contain unique DNA and ribosomes compared to the rest of the cell. And they also have genetic and biochemical characteristics that are more similar to prokaryotes than eukaryotes. This is an 8-minute video. I don't think we'll have time for this. So it's just a general overview of prokaryotes and eukaryotes. So, all right, we have two questions for this chapter. Um, should be fairly simple. Which structure is generally present in both prokaryotic cells and eukaryotic plant cells? Okay, I'll give you like 10 more seconds. Okay, all right, so two people got it correct. The correct answer is B, cell wall. So I'll go over why the other answers aren't true first. So a common answer we saw was C, nucleus. As I mentioned earlier on, a plant cell obviously does have a nucleus. Prokaryotic cells do not have nucleus. They do not have a nucleus. They have what's known as a nucleoid region. And a region is just a very simple region where it contains a small strand of DNA. So prokaryotic cells do not have a nucleus, a fully formed nucleus. They have a nucleoid. So C is unfortunately wrong. Another common answer I saw was D. Prokaryotic cells do not have a mitochondria because one of the main things about prokaryotic cells, they do not have any membrane-bound organelles. A mitochondria, as we mentioned before, has inner outer membrane. It is a membrane-bound organelle. Prokaryotic cells do not have any membrane-bound organelles. So the correct answer, so C and D are out of the question. The correct answer is B. Prokaryotic cells do have a cell wall in order to help protect it, and they also do have, and eukaryotic plant cells also do have a cell wall in order to protect the plant cell. So the correct answer was B. Now we'll go to this question. What does the cytoskeleton consist of? All right, this one everyone got right. It's correct. Microtubules, intermediate filaments, and microfilaments. All right, two more chapters. Chapter 6, biological membranes. So, saturated and unsaturated fatty acids. As we mentioned before, phospholipids extremely important and they make up the bilayer. However, there are differences within fatty acids themselves. Fatty acids can be saturated or unsaturated. What that means, saturated fatty acid means that there are no double bonds in the chain, in the acyl chain. As you see here, you have your hydrophilic head, and then in the acyl chain itself, there are no double bonds between the carbon. Why is that important? If there are no double bonds between the carbon, that means that you can pack it together tightly. So it's just forming a straight line. You can pack these fatty acids together super tightly, and thus these are solid at room temperature. So saturated fatty acids are typically animal fats. So animal fats do not contain double bonds. Because they do not contain double bonds, they are packed together tightly because they essentially just form a straight line and they are solid at room temperature. Below is an unsaturated fatty acid. An unsaturated fatty acid means that it has double bonds. So as you see here, there are two double bonds present. So what is the importance of these double bonds? These double bonds create kinks, which essentially, as we mentioned before, uh, the fatty acid is completely straight when there are no double bonds. When there is a double bond, the fatty acid creates a kink such that, as you see here, it is not perfectly straight anymore. It's either bent to the left, right, etc. So when this bending happens, the kinks prevent packing like you see here. As you see in the top one, the fatty acids are able to be extremely close to each other. Um, okay. The fatty acids are able to be extremely close to each other and in the saturated fatty acid. In the unsaturated fatty acid, the fatty acids are not close to one another, so they prevent packing. As a result, unsaturated fatty acids are liquid at room temperature. So double bonds, liquid, create these bends, and then top saturated single bonds, completely straight. And then there's also mono unsaturated fatty acids and poly unsaturated fatty acids. Mono unsaturated fatty acids are only one double bond. Polyunsaturated fatty acids, as you see here, is more than one double bond. So, what is this yellow thing that may cause a heart attack? This yellow thing is known as cholesterol. Cholesterol is extremely important. The reason for that is that it changes membrane fluidity. Depending on the fatty acid composition of other lipids found in the membrane, it can either increase fluidity or decrease fluidity of the membrane. If you have a lot of cholesterol in your body, it can reduce membrane fluidity to the point where plaques are created. If plaques are created, molecules are not able to properly pass through the membrane or pass through the body as a whole. As a result of that, if you have more and more plaques, it can create blockages, which can eventually cause a heart attack. So this is what's known as the fluid mosaic model. There are several proteins, several lipids, etc., involved in maintaining our phospholipid bilayer. So we'll start with carbohydrates. Carbohydrates are often attached to the outer surface of proteins, forming glycoproteins, or outer surface of lipids, forming glycolipids. Then you have peripheral membrane proteins here. Peripheral membrane proteins do not penetrate the bilayer at all because they don't contain a hydrophobic region. So these proteins, if they don't contain a hydrophobic region, they obviously can't interact with this hydrophobic environment. However, uh, they're still important for if secondary messengers come through the bilayer. So peripheral membrane proteins do not penetrate the bilayer. As you mentioned before, cholesterol molecules are responsible for influencing the fluidity of fatty acids, so it can allow more things to go through or less things to go through. Then there are also membrane proteins. Membrane proteins, some of them are associated with filaments in the extracellular matrix, and these membrane proteins can also be transport proteins as well. And then you have integral proteins. So some integral proteins cross the entire phospholipid bilayer, others only penetrate partially into the bilayer. So all of these combined are responsible for maintaining our cell membrane. So what can actually pass through our cell membrane? The number one thing that can pass through is hydrophobic molecules. Now that makes sense. In order to pass through this hydrophobic region, obviously, hydrophobic molecules will be able to pass through easily. So carbon dioxide, oxygen, nitrogen, or steroids. Extremely small polar molecules can pass through. So polar molecules obviously do not want to interact with the nonpolar region, but if they're extremely small, such as water, glycerol, or ethanol, it will be able to pass through. On the other hand, extremely large polar molecules are not able to pass through, such as actual sugars or amino acids. As a result of that, you need transporters, or they act as secondary messengers, or you need secondary messengers in order to send a response. And then charged molecules are also not able to pass through directly. They need to be pumped through. So this is what's known as passive membrane transport. So you have membrane channels, which are integral. The bilayer is the cell membrane, yes. So the cell membrane, phospholipid bilayer are interchangeable in terms of what you call them. So membrane channels are integral transmembrane proteins. So integral, as I mentioned before, means that it's embedded within the membrane. So the passageway of the channel here is filled with hydrophilic amino acids. The reason why it's filled with hydrophilic amino acids is that it's uh any ion that needs to be passively transported through, if it can't interact with the hydrophobic environment, it needs to interact with the hydrophilic environment. So the internal passageway of membrane channels are hydrophilic so that these ions can pass through. And then any hydrophobic amino acids of the channel membrane protein interacts with the outside. So here, if you can see my mouse, the outside of this channel membrane protein is hydrophobic, so it faces towards the fatty acid tails of the lipid molecules. The inside of the channel membrane protein is hydrophilic so that these ions can go through. So I guess this is a better thing. Here, outside is hydrophobic, inside is hydrophilic so that ions are able to go through. Hydrophilic is hydrophilic is polar, so that's why the ions can pass. Yes. So the reason these ions can pass, like right now, is because the inside of this transport protein is polar. If if it was hydrophobic, then the ions will not be able to transport through. That's the reason why they need a membrane protein in the first place. It's because these molecules are not able to directly pass through the membrane bilayer because it can't interact with the nonpolar hydrophobic region. So these channel proteins have a hydrophilic region in the middle such that these molecules are able to go through. And they also have a hydrophobic region which allows it to be embedded within the cell membrane. So when it comes to ion passive transport, what typically happens is that it's often gated. Gated means that it's usually closed and you need a stimulus molecule to bind to it. A stimulus molecule can be a molecular signal, a pH change, physical pressure, or voltage changes. So a stimulus molecule binds to the site, which then opens up the channel protein, allowing ions to go through. And typically these channel proteins are specific for one type of ion. Now you have carrier proteins. So carrier proteins are responsible for facilitated diffusion. What facilitated diffusion is it involves transporting and binding to the actual molecule itself. So for channel proteins, a stimulus molecule binds to this gate, the gate opens, and then ions are able to freely pass through. Once the gate is open, ions are able to freely pass through. However, for carrier proteins, it physically binds to the molecule itself and then transports it to the inside. And so carrier proteins allow diffusion in both direction. So it can bring glucose from outside to inside, or it can bring glucose from inside to outside. Now we have what's known as active transport. Active transport typically requires energy, usually from ATP, either directly or indirectly. Yeah, carrier proteins help large polar molecules pass through. So when it comes to active transport, the main reason for active transport is that it is moving molecules or ions against their concentration gradient. So here, this is moving with the concentration gradient. Glucose is going from high to low. So it's going from an area of high to low in order to maintain homeostasis, balance it out. This is moving with the concentration gradient. If you're moving something from low to high, that is moving against the concentration gradient. And typically it requires ATP either directly or indirectly. So there are three main types of protein-driven systems involved in active transport. The first one is a uniporter. A uniporter transports one substance in one direction. Then there's a symporter. Symporter transports two different substances in the same direction. So an example of a uniporter is just sodium, potassium, calcium ions can just go inside. Symporter can move sodium and glucose inside, but only in one direction. Once again, for uniporter and symporter, only in one direction. For an antiporter, it can transport two different substances in opposite direction. So it transports one solute into the cell, one solute out of the cell. A common example is the sodium potassium pump, which I'll go over right now. So this is an example of primary active transport. Primary active transport is when ATP itself is used as a source of energy. So, uh-huh. So primary active transport is when ATP itself is used as a source of energy. So this is kind of difficult to understand, so just bear with me as I go over it. Three sodium and one ATP bind to the pump. So in the once again, as we mentioned for active transport, and the sodium potassium pump is an antiporter. So it's moving sodium out of the cell, potassium into the cell. So what this is doing, three sodium and ATP bind to the protein pump. Then the ATP gets hydrolyzed. Once ATP gets hydrolyzed, ADP, so ATP is adenosine triphosphate, which is an A adenosine bound to three phosphate molecules. And then hydrolyzation of ATP converts it into ADP, adenosine diphosphate, which is an adenosine bound to two phosphates, and then it has one phosphate free flowing. In this case, once ATP is hydrolyzed, ADP leaves, and one of the phosphates from ATP is bound to the membrane protein. Because it's bound to the membrane protein, it changes its shape. So first, the protein pump is facing inside the cell, obtains three sodium, and then bound to ATP. ATP gets hydrolyzed into ADP and the phosphate. Phosphate group still binds to the protein, and it's now facing extracellularly. It's now facing outside the environment. It is releasing sodium out the environment. Now, two potassium bind to the cell. Two potassium from outside the cell bind to inside the cell. Once two potassium from outside the cell binds to inside the cell, eventually not phosphorylation, dephosphorylation happens, which means that phosphate is released from the carrier protein. Once phosphate is released from the carrier protein returns back to its original shape, releasing two potassium ions into the cell, and once again exposing these binding sites for sodium to come in again. So to go over it really quickly one more time, sodium and ATP bind to the pump inside the cell. ATP gets hydrolyzed, ADP leaves, phosphate still binds to the protein. Now the protein faces outside the cell. Three sodium from inside the cell leaves the cell now. It opens the binding sites here for two potassium outside the cell to bind to the pump. Now 2 potassium is bound to the pump. Now phosphate gets dephosphorylated, which means phosphate leaves the protein pump so that the pump can return to its original shape, releasing 2 potassium inside the cell and exposing these three binding sites for sodium again. So now we're going to talk about endocytosis. We talked about endocytosis a little bit a few minutes ago when we referred to phagocytosis. So endocytosis essentially bring materials from outside the cell inside the cell. Endo, inside. Exocytosis is the opposite. It's moving stuff outside the cell. So typically what happens, you have phagocytosis, which is when you take in larger substances such as bacteria inside the cell. So this is through membrane invagination, where the internalization of molecules of other substances are brought into the cell, and then the membrane closes in on itself. So the plasma membrane folds inward, engulfs the material, and forms a vesicle. So that's phagocytosis. Pinocytosis is the same exact thing. It works through membrane invagination. However, the molecules are much small. However, the vesicles, this vesicle here that's surrounding the bacteria, the vesicles are much, much smaller because it's taking in fluids and solutes. Then you also have receptor-mediated endocytosis. So receptor-mediated endocytosis is highly specific. It depends on receptor proteins here. So it depends on receptor proteins here, which are integral membrane proteins, so they're inside the membrane, and they bind to specific substances. Once the specific substance is bound to bind oh sorry, once a specific substance actually binds to the receptor protein, membrane invagination happens once again. Now you have a coated vesicle. So the sites of internalization, when it starts to fold in on itself, are known as coated pits. And these coated pits have specialized proteins known as clathrin, which help coat the vesicle once once the substance binds to the receptor. Exocytosis is essentially the opposite. It expels materials that are indigestible, for example, via vesicles. Now, what happens? These vesicles fuse with the cell membrane, releasing the contents inside outside. Other examples of exocytosis are hormones or neurotransmitters, which you'll learn about in when you talk about the nervous system. Neurotransmitters are also released through exocytosis. So now we're going to talk about cell junctions. So cells form junctions among themselves in a tissue, and there are three main types of junctions: tight junctions, desmosomes, and gap junctions. Tight junctions are commonly found in epithelial cells, which are skin cells, and the plasma membrane, sorry, the plasma membrane between adjacent cells are extremely tightly linked together. So because they're tightly linked together, there's not much that's able to pass through between the cell. So it restricts any migration of membrane proteins and phospholipids and helps make sure substances cannot move through the intracellular space between adjacent cells. So tight junctions prevent movement of molecules within cells. Then there are desmosomes. Desmosomes weld adjacent cells together, and after they weld adjacent cells together, they promote cell interactions with each other. So because the adjacent cells are obviously right next to each other, they help promote cell-cell interaction, and they also prevent the breakage of cells during stretch. So it helps make sure that these cells aren't breaking apart and they're still tightly weld together. Lastly, there are gap junctions. Gap junctions are typically found in cardiac cells, and they're also found in neurons as well for electrical synapses. So with gap junctions promote more communication between cells. They're made up of protein channels known as connexons, which span the plasma membrane of two adjacent cells. And as you see here, it protrudes slightly into the cytoplasm. And then connexons, these gap junctions are important because signals are sent into the gap junction. Signals are sent into the gap junction in order to pass it to the adjacent cell. All right, first question. Which statement about membrane proteins is not true? Okay, so we have a lot of differing answers. So the not true questions are usually the most difficult questions because for like when the question is which statement is true, you just have to look for one thing that sounds correct and then that's the answer you choose. However, when it comes to which statement is not true, all five of them can sound correct, which is why it's the hardest question. But the correct answer for this is A. They all extend from one side of the membrane to the other. So the reason A is wrong is because there are peripheral membrane proteins here. So peripheral membrane proteins do not penetrate the bilayer at all. And as we mentioned before, for integral membrane proteins, some of them can cross the entire phospholipid bilayer, but others penetrate only partially into the bilayer. So the remaining four answer choices are true, but A was not true. So the correct answer was A. So I know C, you could think, oh, why is C correct? But that's because as long as the integral membrane protein here, as long as there is an integral membrane protein within the phospholipid bilayer, it can move laterally within the membrane because they already has an area that's exposed to the hydrophobic region. So it can move laterally, which means across the phospholipid bilayer easily into a new spot so that more binding, etc., can happen. All right. Um, Primary and secondary active transport. I know we didn't talk about secondary active transport, but let's see if you guys can get the answer through Dr. Fat's lectures. All right, yeah, everyone got it correct. Primary and secondary active transport both move solutes against their concentration gradients. So, only primary active transport uses ATP directly. Secondary active transport, what it does is, if we'll go here. So this is an example of primary active transfer, right? You have sodium out of the cell, potassium into the cell. Secondary active transport is whenever an electrical chemical gradient happens, it can use that electrochemical gradient to move solutes against this concentration gradient. So here, primary active transport moves sodium out of the cell using ATP. Now that there's more sodium out of the cell, it creates an electrical chemical gradient such that because there's more sodium out of the cell, that can help push other ions into the cell. So there's a sodium glucose concentration gradient. So that's just an example. Having more sodium out of the cell can help push glucose into the cell. So secondary active transport uses an electrical chemical gradient in order to push other solutes against the concentration gradient, while primary uses ATP directly. All right, now we're in the last chapter, cell signaling and communication. I'm gonna drink a little bit of water first. Sorry. All right, last chapter. So this is what's known as a common signal transduction pathway. A signal molecule binds to a receptor. A receptor can then activate another signal transduction molecule. What this signal transduction molecule does, it can create short-term changes. So is it the sodium ions that push the glucose? So the sodium ions creating that gradient, creating that difference in electrical voltage, pushes the glucose in the cell. The sodium ion itself does not necessarily like sodium itself doesn't act as like a transport protein, but because now there's more sodium out of the cell, it's creating an electrical chemical gradient that can push other ions or molecules within the cell for secondary active transport. All right, yeah. So sorry, signal transduction pathway. Signal molecule binds to a receptor. Receptor can then activate another signal transduction molecule, which causes both short-term changes and long-term changes. Examples of short-term changes, it can activate or inactivate certain enzymes. Examples of long-term changes, it can alter DNA transcription as a whole, so that any future cells, once it replicates, has that DNA transcription. So there are different methods of cellular communication. You have autocrine, juxtacrine, paracrine, and endocrine. Autocrine cells, autocrine signaling is when the chemical signal affects the signaling cell itself. So here, this signal goes back on itself and then affects it. So autocrine, auto, it's affecting itself. Juxtacrine is that chemical signal is affecting directly adjacent cells. So as you see here, these two cells are directly adjacent to each other. So this signaling here, where this signal binds to this cell, this signaling is known as juxtacrine. Juxtacrine adjacent cells. Paracrine signaling is when you affect neighboring cells. So the signal from this receptor molecule or this secreting molecule or cell binds to this receptor molecule here. So because it's nearby, it is known as paracrine. And this target cell does not have any receptors, so it's obviously not going to respond to the cell and not going to bind to the signal. So once again, autocrine is the chemical signal binds to itself. Juxtacrine binds to adjacent cells. Paracrine binds to nearby cells. And lastly, you have endocrine. Endocrine signaling is basically the chemical signal is delivered to a very distant cell or distant target organ, target molecule, etc. So exocrine is or endocrine is very distant chemical signaling. Is it neighboring and adjacent synonyms of each other? No. So adjacent means it's directly attached to. Neighboring just means like a house, the house right next to you could be your neighbor, but the house like two houses down could also be your neighbor, or three houses down could also be your neighbor. Neighboring cell, just think of it as a cell that's nearby. Like it's not directly attached to the secreting cell, it's just nearby that cell. While for juxtacrine signaling, the adjacent cell, it is directly attached to that cell. So juxtacrine directly attached to paracrine is it's nearby. And then endocrine is, like I mentioned, delivered to a target, distant target cell, target organ, etc. So you have what are known as cytoplasmic receptors as well as transmembrane receptors. So cytoplasmic receptors interact with lipid-soluble signaling molecules. So for example, cortisol. Cortisol is lipid soluble, which means it can go through the cell membrane really easily. It goes through the cell membrane, now it's inside the cell, inside the cytoplasm. Once it's inside the cytoplasm, it can bind to a receptor molecule within the cytoplasm itself, and then that causes further changes later on. So this is what's known as a cytoplasmic receptor. Receptor inside the cytoplasm binds to a molecule, a signaling molecule that can transport directly through the membrane. However, certain molecules are not able to pass directly through the membrane, such as your polar molecules or charged molecules. These polar signals bind to what's known as a transmembrane receptor. Transmembrane receptors are integral membrane proteins. They're obviously within the actual membrane itself. So once they bind to the receptor, these receptors cause changes. You have different types of transmembrane receptors. You have ion channel receptors, protein kinase receptors, and GPCRs. So first we're going to talk about agonists and antagonists. So binding of a ligand changes the configuration and it activates a certain receptor complex. So agonists, what they do is they mimic the action of the natural ligand, which means whatever the natural signaling molecule is, if you have an agonist, it can mimic the action and cause the same signal to occur. Antagonists prevent the binding of the natural ligand such that the signal does not occur anymore. Caffeine and adenosine the is a great example of an antagonist. Caffeine is a natural antagonist of adenosine. What adenosine does, adenosine typically binds to a receptor, and then adenosine is responsible for sleep promoting properties. So adenosine causes you to fall asleep. However, what caffeine does, caffeine blocks the binding of adenosine to its receptors. So caffeine is an antagonist. It's able to bind and make sure that adenosine is able to block the binding of adenosine to its receptors. Thus, caffeine reverses any sleep promoting properties and instead promotes wakefulness and alertness. So that's why caffeine itself does not necessarily promote wakefulness. It's the fact that caffeine blocks adenosine to bind to its receptors that promotes wakefulness. So here we're going to talk about ligand-gated ion channel receptors. Um, As we mentioned in, I believe, the last chapter, it's very similar. You have a signaling molecule binds to the channel membrane protein. Once the signaling molecule binds to the channel membrane protein, it opens the gate up, allowing ions to go through. So this is an ion channel receptor. Signaling molecules open or close receptor channels. So in this case, it's opening. Other signaling molecules can close it. So there could be gates that are typically open. A signaling molecule comes in and it does the opposite, it closes it. So now we're going to talk about phosphorylation as a transduction mechanism. So what does that mean? So typically what happens, as we mentioned before, ATP is adenosine triphosphate, contains three phosphate groups. ADP, adenosine diphosphate, contains two phosphate groups. Sometimes there are things known as phosphorylation, where you add a phosphate onto a protein in order to either activate it, inactivate it, but you're changing its shape and thus changing its function. So you have protein kinases. Protein kinases specifically, are responsible for phosphorylating other proteins. So a phosphate is transferred from ATP. So ATP, as you see here, has three phosphates. Now, a phosphate is transferred from ATP onto a protein. Now it has two phosphates, ADP. So now that a phosphate is transferred onto a protein, that protein becomes phosphorylated as an altered shape and function. So here's an example for an insulin receptor. Insulin can bind to an insulin receptor, and then sometimes certain molecules are responsible to phosphorylate it, like we saw here. A protein kinase can sometimes be responsible to phosphorylate it, but sometimes you can also have receptor kinases phosphorylate itself. So this is known as autophosphorylation. So insulin binds to a protein kinase receptor, then autophosphorylation occurs where the receptor kinase is able to autophosphorylate, which means add phosphates onto itself without the help of any other molecules. Now that there's phosphates here, an insulin response substrate can come and then take a phosphate from the kinase. Once the substrate is phosphorylated, it causes cellular responses within the cell. So insulin normally this kinase is inactive, but insulin binding to it activates it, causing autophosphorylation, causing a substrate to be phosphorylated, and then allowing for cellular responses to occur. So this is what I mean by phosphorylation can occur in both ways. So ATP becomes ADP, and then also GTP for G protein coupled receptors can become GDP. A kinase phosphorylates it, which means it adds a phosphate onto a protein. This can, depending on the protein, it can either be active or inactive. And then, in order to return it back to normal, phosphatase is responsible. Phosphatase is an enzyme. Phosphatase gets rid of a phosphate from protein and then dephosphorylates it, making it the opposite, either active or inactive. So now we're going to talk about G protein coupled receptors. G protein coupled receptors are a bit hard to understand at first, but I'll go through it as best as I can. So you have a signaling molecule here. Signaling molecule binds to a G protein linked receptor, which nearby is a G protein. A G protein is inactive when it binds to GDP. So when it binds to GDP, it is inactive. Signal binds to the receptor. That receptor phosphorylates the G protein, causing it to bind to GTP. So here's an example through smell. Your ligand, which is your receptor molecule, is an odorant, a smell. Receptor is obviously a G protein linked receptor. So odorant, a smell, binds to the receptor. That receptor then activates your G protein here, which converts it to GTP. So a G protein coupled receptor has three subunits. One, two, three. So it has three subunits, and then one of the subunits here, so you see here has three subunits. One of the subunits disassociates. Okay. One of the subunits disassociates from the G protein linked receptor and then, oh, sorry, and then binds to an effector protein. This effector protein is now activated. So in this case, the effector protein is adenylyl cyclase. So adenylyl cyclase is normally inactive. Now the G protein is active. The alpha subunit that's bound to GTP. So the alpha subunit then dissociates from the G protein coupled receptor, binds to the effector protein, which is adenylyl cyclase. This then causes second messengers to occur. What are second messengers? Second messengers are, as is the name, it's a secondary messenger that causes a secondary response to occur. Adenylyl cyclase converts ATP into CAMP. And what does CAMP do? CAMP binds to this channel, and then the channel causes calcium and sodium to cause an influx. So it goes within the cell, and that sends a signal to the brain. So your original method or your original outcome is ligand binds to receptor. Receptor activates G protein. The G protein typically has GDP activated with GTP. One of the subunits with GTP then activates an effector protein. Effector protein becomes activated, causes a secondary messenger to occur. So in this case, it converts ATP to CAMP. Secondary messenger then causes whatever function it needs to happen. In this case, it binds to a channel, allowing calcium and sodium to go in. So certain pathogens can interfere with G protein functions. So here you have cholera. What cholera does is that, so as I mentioned before, when the G protein is activated here, GTP activates it. Eventually, the subunit GTP gets hydrolyzed into GDP so that everything can return back to normal, right? Everything eventually has to come back to normal because you don't want this to be activated forever. What cholera does is that it binds to the G protein right here. So once the G protein is activated, it binds to the G protein and prevents the hydrolysis of GTP. So typically what happens, the GTP becomes GDP so that this subunit can return back to normal and everything starts at the beginning again. Cholera binds to the GTP protein binds to the G protein and prevents any hydrolysis of GTP such that the G protein is persistently activated and the effector protein is also persistently activated. So there's continued cellular responses. And then the opposite can also happen. So for cholera, the problem was that you were having too many cellular responses. Pertussis toxin, which is responsible for whooping cough, can cause the opposite effect where you have zero cellular responses. What it does is that it binds to G protein and prevents its interaction with the receptor and the release of GDP into GTP. So pertussis toxin can bind to the G protein here once it's inactivated. Typically, it exchanges GDP for GTP and becomes activated. However, pertussis toxin binds to the inactive G protein and keeps it permanently inactivated, so no cellular signal response can occur. So we're talking about secondary messengers. Secondary messengers are part of the signal transduction cascade within the cytoplasm. As mentioned before, their main point of secondary messengers is they help alter the activity of other enzymes and other channels and proteins within the actual cell that the original messenger is not able to go through. So they participate in that signal amplification process. Common examples is CAMP, CGMP, DAG, IP3, calcium, and nitric oxide. Um, So in this case, you have acetylcholine. Acetylcholine activates a G protein dependent IP3 DAG transduction system in endothelial cells. So here acetylcholine binds. You can't see the G protein here, but there's a G protein that then activates IP3. What IP3 does is that it makes sure calcium can binds to this calcium channel, makes sure calcium can go inside the cell. Calcium can then bind to nitric oxide synthase, and what nitric oxide does is that it stimulates production of CGMP in your smooth muscle cells, which then relaxes it. So this whole process is just an example of how many secondary messengers there could be and just how long the process can be. This is what they mean by signal amplification. An original signal here can cause a secondary messenger. This secondary messenger can cause further signals to occur until the final end product happens. So this is just one more time. Epinephrine binds to a receptor. G protein gets activated, exchanges GDP for GTP. GTP alpha subunit dissociates and then binds to an effector protein. In this case, it's activating adenylyl cyclase. Adenylyl cyclase is activated, converts ATP to CAMP. CAMP, as we mentioned before, is a common secondary messenger. Here it has two functions. Glycogen synthase becomes inactive, which means your body is not forming more glycogen, and it activates protein kinase A. Then more signals. Protein kinase A activates phosphorylase kinase. Phosphorylase kinase activates glycogen phosphorylase, and then glycogen phosphorylase converts glycogen into glucose so that our blood glucose levels can rise. So once again, this is just another example of how many signals can lead to further signals, and they can, as you see here for CAMP, CAMP can activate a molecule and inactivate another molecule, so it can work in both ways. So how are cells regulated? We're almost done. So cells are regulated through several methods. One, protein phosphatase. As you mentioned before, phosphorylase and protein kinase can phosphorylate certain molecules. In this case, it's activating the enzyme. It can also inactivate it, but in this case, it's activating the enzyme. Protein phosphatase gets rid of the phosphate, returns it back to its original state. Same thing with GTP. As you mentioned before, the G protein, once it's activated, it'll go to the effector molecule. You don't want the effector molecule to be activated permanently because that means there's cellular responses continuously. So GTPase comes in, converts GTP into GDP, so it goes back to its original state. And lastly, so these are self-regulatory mechanisms, basically. A self-regulatory means the mechanism itself, it has regulations in its own body in order to help switch it on and off. And then lastly, you have metabolism of signaling molecules. So as you mentioned before, CAMP is a common secondary messenger. If you have too many secondary messengers, phosphodiesterase comes in to break the ring of CAMP, convert it into AMP, and then it now CAMP is inactive. So it can't act as a secondary messenger anymore. All right, last two questions. A nonpolar ligand such as a steroid hormone usually binds to blank. Oh, I was muted. I thought I was talking. Sorry about that. Here. So the correct answer was A, intracellular receptor. Thank you for letting me know. The reason it's A is because a nonpolar ligand, what does nonpolar mean? It can it interacts with the hydrophobic environment of the phospholipid bilayer. So because it can interact with the tails, the fatty acid tails, it can directly diffuse through the cell membrane. Because it can directly diffuse through the cell membrane, it has to bind to a receptor that's on the inside of the cell, which is an intracellular receptor or a cytoplasmic receptor, as we went over before. So the correct answer for this is A. So now for this question, why do some signals trigger secondary messengers to activate target cells? All right, so we have a few different answers. So the correct answer is D. The first messenger cannot cross the plasma membrane. So the reason it's not C, so the first messenger does not actually bind to many types of cells. The first messenger just binds to whatever receptor it has. And then that receptor, like for example, a G protein coupled receptor, it just binds to the G protein coupled receptor, and that receptor then activates further the G protein, which then binds to whatever other protein, etc. So the first messenger, it can't cross the plasma membrane because typically it's a large polar molecule. So in that large polar molecule, such as here, epinephrine. Epinephrine is a large polar molecule. It only binds to epinephrine receptors themselves. This can't cross through the membrane, so it needs to bind to a receptor on the membrane, which can then allow epinephrine's original purpose. It can then allow secondary messengers to carry on its purpose within the cell. So yeah, because the first messenger cannot cross the plasma membrane, it triggers secondary messengers to then activate whatever target cell it requires. And then, okay, so yeah, that's everything. Before you guys leave, if you guys did not fill out the attendance form, please fill out the attendance form because that's not for Dr. Fath or Professor Garofalo. That's for us to know, obviously, that you guys need more review sessions in the future. It would be really helpful for us to know that, this went well. Same thing with the feedback form. Please, please, please fill out the feedback form. That way I also know what I need to improve on or how well the session went, what you guys also want in the future review sessions. So please fill out both. I'll keep this up I'll keep this up here. I'll also talk about general advice for the test. The attendance form is on the right, so the attendance form is the QR code for the right. Feedback form is the QR code for the left. So please fill out both. It'd be really helpful for me and the learning commons. So, a general advice for the test. If you noticed from my questions are fairly specific. There aren't many like story type questions where you'll have to read a passage based off the passage, you'll have to infer what the answer is. To be quite frank, if you have looked at the sample exam is typically much easier than the actual real exam, but the sample exam itself, most of its questions are very direct, super straightforward questions, similar to mine. So, what this means is that you're not going to have much opportunity to guess, like make a best educated guess on what the answer is. So in that case, you're really gonna have to spend a lot of time actually just making sure that you're able to know the answer offhand and like memorize essentially each slide. So the way the exam is formatted is there's gonna be anywhere from 8 to 14 questions per chapter, and the test is thankfully in order. So essentially the first 8 to 14 questions is chapter 2. Next 8 to 14 chapter 3, so on and so forth. Typically anywhere from 50 to 70 questions on the test. Like I mentioned before, the questions themselves are, for the most part, very direct, very straightforward. So you're not really gonna have an opportunity to make an educated guess. It's either you know the answer or you don't know the answer. Um, Number two, and only multiple choice, yes. I would recommend studying from the slides. The reason I recommend studying from the slides is because I've seen Dr. Fat and Professor Garofalo, what they do, they genuinely just look at the slides and then start making questions. They look at each slide and make questions. What that means is that any slide can have a question on the exam. And so I also would not recommend reading the textbook because of that. Dr. Fat and Professor Garofalo do not look at the textbook at all when it comes time to making questions. They solely look at their slides. So the textbook, as well as today's review session, should just be used as supplementary materials for the actual review of the slides itself. Essentially, if there's something on the slides that you're confused about, let's say there's like a whole image that you're let's say there's an image and there's not a lot of wording going over what the image is. So in that case, you can use the textbook or use this video later on and then to learn more about what that image is saying. However, once again, it should only be a supplementary material. Every single question will be based on the slides itself, and that's it. So I really would recommend studying from the slides. I don't recommend solely studying the sample exam. The sample exam itself, like I mentioned, it is much easier than the actual exam. However, the sample exam does give a good idea of the type of questions you're expected to understand. Like I mentioned, these questions are very direct for the most part. There will be very few questions where you can make educated guesses. For the most part, it's what is true, what is not true, what is the function of XYZ. If XYZ, like let's say a protein, if the G protein is not activated, what will happen? Or like cholera, like we talked about cholera. If cholera blocks the G protein from being deactivated, what will happen? What's gonna happen? There's gonna be more cellular responses. It's like very direct questions like that. So try your best to make sure you know every process. Um, Yeah, that's pretty much it. I think you guys do have more than enough time, in my opinion. It is only two, three, four, five, six, seven. It is only six chapters. You guys have the rest of today, Monday and Tuesday is off, Wednesday, and then Thursday it's evening as well. So you guys have essentially 4 days to go over six chapters. You could easily do two chapters a day. If like today's your first time ever studying or even looking at this material, don't worry, you still have 4 days to go over six chapters, which I personally believe is more than enough time. If you go over two chapters a day for the next 3 days, and then another day where you just go over everything at once, you should be ready for the exam. Another thing, please don't underestimate biology 105 in the exam. I'm sure you guys have heard stories about 105 class averages, etc. All of it is true. A lot of students come in because obviously it is their first semester. A lot of you guys are freshmen. A lot of students come in thinking, oh, it'll be like high school biology, it'll be very general. No, the questions will not be very general. The questions will be specific. Please make sure you take the proper time and effort to actually study each slide and go over, make sure you know everything that's applicable. Because that's basically what the exam will be. Um, and yeah, like I mentioned, please study from the slides. You guys do have enough time. If you do two chapters today, two chapters Monday, two chapters Tuesday, Wednesday could be a all-around review session day, and then Thursday as well. I think you guys will be good to go. Another thing, don't disregard lab as well. I've said this to a lot of my other students. Please don't disregard lab. The reason I say that is because biology is the only class where lab is not a separate class. In chemistry, physics, etc., lab is a one credit course in a separate class. So, lab is a one credit course and it counts as a separate class. It doesn't count as your overall grade. In biology case for 105 and 106, lab is worth 40% of your grade. If you include homework, which I hopefully you guys are all doing on time, lab plus homework is half of your grade. So if you know that you're not the best test taker, obviously do your best to make sure you reach a certain like level or point on the exam, but lab and homework can easily boost your grades up. If you know that you're much better at writing reports, taking your time on certain things, then lab and homework can easily boost your grade up a whole point or a whole like grade range. I know there are hundreds of kids that have gone from C's to B's, B's to A's, purely due to their lab and homework, because that is half of your grade. So please do not neglect lab and homework. So if you know you're not the greatest test taker, you can definitely still do better through lab and homework. I'll go over the questions now. Which room are you taking your exam? The room you're taking your exam is the big lecture hall. I think it's 171 if I'm not mistaken. If someone can correct me on that, is 170, yeah. So this is the main lecture hall. However, I know there are certain lab classes that have it in a separate room. If anyone's taking Sophia's lab, I believe she has hers in a separate room, so please keep that in mind. Because some of you guys may be sent to her room. Another thing, please come in on time. Um, you know, it is a 6:30 class, but please come in on time. It'll be like the test will happen like within 5 minutes. No, Professor James Carpino is not new. He's been here for several years. Um, so please come in on time. Yeah, he's been here for several years. So please come in on time because you'll also need to find seats. And then if there are no seats, you'll be forced to sit in the front. And if there are no seats in the front, you'll be forced to go to the other room. So please come in a bit early if you can. Can we access the recording? Yeah. So the recording will be on the Queens College Learning Commons YouTube. So it won't be in a mass email. What'll happen is that here, Queens College Learning Commons. So if you search that up on YouTube, you should find it. I have an exam one review session from last semester already up. So by the time this gets posted, you can access the one from last semester, which I believe went over the same exact chapters. So you can access that review session as well. So just search up Queens College Learning Commons on YouTube. You'll find the bio exams, and I have it for every single one. So you should be able to access it through that YouTube. Yeah, post this recording will be posted on YouTube. We will sit for exams in three different classrooms. I'm not sure if it'll be three different classrooms. Obviously, I'm not Dr. Fath, I'm not the professor himself, so I'm not able to tell you exactly. I know there's guaranteed to be two rooms. The main room he always does lecture in, and then another room for certain lab courses. I don't think there will be another room. If there is, then Obviously, Dr. Fath and Professor Garofalo will tell you themselves in they'll tell you themselves in Brightspace. But for most students, it should be within the main lecture hall that he normally lectures in. So the previous exam he posted, so like I mentioned, that's the sample exam. The sample exam is much simpler or easier than the actual exam. However, it is the same style. What I mean by same style is the directness of the questions. That's what you can expect on the exam itself. So the style of questions he goes over the exam is the style of questions that'll be on the actual test. No, you guys are not seated in alphabetical order. It's first come, first serve, sit wherever. Of course, if you come late, then you'll be forced to seat in certain areas. Recording posted on the YouTube. If you scroll up, you should find the YouTube link. Um, are there any resources we can use to study or do practice questions beside the sample cues? When I took the class and what I recommend, like all my students, I always recommend just studying from the slides. That's it. Slides and then sample cues and then just more and more slides. Um, yeah, honestly, that's what I would recommend. Do I have any tips for lab quizzes? So I don't have much tips for lab quizzes because each the lab quizzes aren't departmental. It's dependent on each lab professor. So one lab professor can do 20 lab questions. Another lab professor can do five. One could do multiple choice, another could do short response. It's purely dependent on who your lab instructor is. So unfortunately, I don't have any tips for that. The only tips that I do have is that, you know, it it'll probably be just based on like the main topics of the lab. It's like the lab summary that you read beforehand. It'll probably be mainly based off the lab summary and also the results. The results are typically the main thing. Like, I know for like the pea glow lab, what causes the bacteria to glow, what caused the bacteria to not glow, etc. It'll be like mainly based on the lab. Can I upload the slides onto ChatGPT or Quizlet and study off of that? Sure. I mean, if that's what helps, I know a lot of students do that. They upload the slides because you are able to upload the slides. So they just download it, upload it, and then ask it to give direct questions. Give me direct questions from each chapter. And then maybe you could also post his sample exam too. Give me direct questions similar to the sample exam but more difficult. That could definitely be very helpful to you in terms of studying because yeah, it'll purely be based off the slide size. If you have every slide, I believe if you truly do have every slide like down to a T, you should do very well on the exam. Yeah, if anyone has any more questions, feel free to respond in the chat. Please fill out the feedback form. It'll be very helpful for me to know what I can improve on and how this session went as a whole, especially in the future. Please fill out the attendance. No worries. I'll see you guys on Thursday as well. Um, I'll be proctoring during that time, so I'll see you guys Thursday. Yeah, have a great day, everyone. Thank you guys for your kind words as well. All right, bye. Oh yeah, let me stop. How do I stop recording? Pause.